Introduction, and good afternoon, everyone. Thank you for coming to my talk, although it overlaps with the EuroCup final, so I hope you will enjoy the talk. So, as Jian Li introduced, my name is Xin Gao. I'm currently an associate professor of computer science in Computational Bioscience Research Center at KAUST in Saudi Arabia. So this talk is about our efforts on developing a machine learning framework that simultaneously explores protein sequence space and protein three-dimensional structure space in order to solve the distant homologue detection problem, which can consequently improve the state of the art for protein structure prediction. So in the field of bioinformatics and computational biology, there are some problems that do not need much introduction, and protein structure prediction is one of them. So the input and output for protein structure prediction is very simple, where the input is any arbitrary given query sequence of a protein, and the output is the predicted three-dimensional structure. So despite of this very simple setup, protein structure prediction has been one of the oldest and one of the most challenging problems in computational biology, and yet it is an indispensable step in understanding protein functions. So during the past 60 years, hundreds of methods have been developed for protein structure prediction, and they can roughly be characterized into three classes. The first one is physics-based methods, which are usually referred to as ab initio methods. So the idea is to find an energy function and then search the conformational space in order to find a structure that corresponds to the global minimum of the energy function. And apparently, such methods will suffer from the inaccuracy of the energy function as well as the huge conformational space. And the second type of approach turns to be better, and they are called knowledge-based methods, which are often referred to as homologue detection-based methods. So the underlying assumption is that two proteins, they are homologues if they share same ancestry, and thus they should have similar three-dimensional structures. So depending on whether the homologue has high sequence identity to the query protein, we can have two versions of the problem: close homologue detection and distant homologue detection, where close homologue detection can be easily solved by any sequence alignment methods, whereas distant homologue detection still remains to be one of the main open problems in protein structure prediction. And usually people need to involve threading technique, which tries to align the query sequence to the three-dimensional structure of the templates in the PDB database and see how well that can fit. And of course, there are many, many existing methods that try to combine the strengths of other methods. So if we zoom out a little bit and try to consider at the very high level what kind of information has been and has not been used by all the existing methods, then I'm drawing you some cartoon illustration. Suppose I put all the protein sequences in a space, and in order to do that, I need some sequence similarity matrix, and we will talk about that later. But for now, just assume we have a sequence similarity matrix. And then I can form this protein sequence space, which also contains my query sequence. And similarly, if I have a structure distance matrix, then I can define a three-dimensional structure space that contains all the protein three-dimensional structures. Then at the high level, the sequence alignment-based methods either try to align or measure the similarity between the query sequence with every single other sequence in the sequence space, or some of them try to align the query sequence with a predefined very small subset of sequences in the sequence space, which are often referred to as protein families. Whereas the threading methods try to estimate the distance from the query sequence to every single structure in the three-dimensional structure space. Last year, Xiele's group at City University of New York, they developed a method that clusters all the three-dimensional structures in the structure space into clusters or families. And then they start from the query sequence, use some random walk technique to traverse the structure space. However, to our knowledge, none of the existing method has explicitly and directly incorporate the entire topology and network information of the entire sequence space and that of the structure space. And that's the motivation of our work. So we wanted to develop a machine learning framework that can simultaneously and explicitly incorporate the entire sequence space information and structure space information, and we want to see whether that can help improve protein homology detection and consequently protein structure prediction. So based on that idea, we developed this machine learning framework called CMsearch, which denotes cross-model search. So as I said, at the beginning, I have the sequence space and the structure space. And I have some initial incomplete one-to-one mapping between the two spaces. And that can be as simple as the same protein has a sequence representation and a small portion of them have a structure representation in the structure space. So it's an incomplete one-to-one mapping. And what I want to do is that I want to simultaneously consider the intraspace information as well as the interspace information in order to learn a more complete and hopefully more accurate many-to-many mapping between the protein sequence space and the protein structure space. And hopefully the edges I learned represent the homologous relationship. So mathematically speaking, suppose I have a protein sequence space denoted as S that contains n sequences. And suppose I have a structure space denoted as T that contains m structures. N is not necessarily equal to m. So the final many-to-many mapping matrix I want to learn, I denote as X, which is of dimension n times m. At the beginning, I have some initial guess of X, which I call that X prime, which is simply a binary matrix, where each entry can be either 0 or 1. 1 means the sequence and the structure corresponds to the same protein, and 0 otherwise. So my objective function contains the following terms. First of all, I request my finally learned matrix X cannot be too different from my initial matrix X prime. And for that, I put a quadratic loss term here. And secondly, if I have a sequence similarity matrix in my sequence space, and again, let's assume I have that for now. So if two sequences, Si and Sj, they are very similar to each other, then I would assume that their corresponding mapping vectors to the structure space should also be similar to each other. Otherwise, I will penalize that. So for that, I put a weighted quadratic loss term here to penalize that. And you can show that this is actually the graph Laplacian of the protein sequence space. And similarly, if I have a structure similarity matrix to define the structure space, then if two structures Ti and Tj are similar to each other, then I would say that their corresponding mapping vectors to the sequence space should also be similar to each other. Otherwise, I penalize that. So this turns out to be the graph Laplacian of the structure space. So finally, my objective function is a linear weighted combination of the three loss terms. And of course, I want to minimize this objective function. And to minimize that, you just take the derivative with respect to x. And you can easily show that this is a Sylvester equation, which gives you a unique solution. However, solving this equation exactly is very time-consuming. So in practice, we adopted an algorithm developed by Zhou et al. In 2004, which decomposes this objective function into two sub-problems, and then iteratively solve each sub-problems and combine their results together. So I won't get into the technical details. If you're interested in, please come to our paper. Now, what's needs to be clarified is we haven't decided what sequence matrix we use to define the sequence space and what structure matrix we use to define the structure space. So the state of the art to define sequence similarity is based on profile ideas because they can capture the evolutionary information much better than the individual sequences. And there are three standard ways to represent protein family profiles. And those are position-specific scoring matrix, PSSM, hidden Markov model, or Markov random field. So in order to get PSSM, you can easily run SciBlast, and then you get this PSSM about a protein family, and then you can easily align two protein sequences together by aligning their PSSM together. Or alternatively, you can build hidden Markov models, which is supposed to capture better evolutionary information than PSSM, and then you align hidden Markov models together. And recently, people have been using Markov random field to represent protein family profiles. And Markov random field can theoretically capture long-range residual interactions. And for that, it's supposed to be better than hidden Markov model. So in our work, we tried each of these three sequence similarity matrix one by one. We didn't combine them, and I will show you results later on. In order to define the protein structure space, we just used the standard TM score. And we chose TM score because it's one of the most widely used methods, and it's independent from the total number of residues, and the value always ranges between 0 and 1. So to test our method, we chose standard PDB40, in which every single pair of sequences share less than 40% sequence identity. And of course, we do some filtering, which ends up with more than 8,000 proteins. So we will use every single one of these 8,000 proteins in turn as our test data. So this is a pretty large experiment in terms of homology detection and protein structure prediction. And we did a 32-fold cross-validation on this entire PDB40 dataset. And as I said, in order to define our sequence space, we chose three methods respectively. Those are BLAST, which can give us PSSM alignment, HH-search, which can give us hidden Markov model alignment, and MRF align, which can give us MRF alignment. And to define the structure space, we just use TM align, which gives us TM score. So the first experiments we did is we want to see, comparing with the original sequence similarity search methods we use, how much improvement we can gain. So for that purpose, we asked the three sequence similarity search methods to return the top K predictions for each target. And then we measure the recall and the precision among the top K predictions comparing to the ground truth homologs. And the blue curves shows those three methods' performance. And the red ones are the corresponding version of CMsearch that uses these corresponding sequence similarity search to define our sequence space. And we run cross-model learning, and we finally return another top K list, and we compare with the ground truth. So you can see that, as expected, BLAST performs much worse than HH-search and MRF align. However, no matter which sequence similarity search you use, our CMsearch can always dramatically improve both recall and precision. And more importantly, no matter which input sequence similarity search you use, we can sort of improve the performance to similar levels, which is sort of suggests that CMsearch is insensitive to the choice of sequence distance metric. And next, we compare the homology detection performance of CMsearch with three state-of-the-art methods for homology detection, including HH-search, which is in black, I don't know if you can see it, and RaptorX, which is in blue, and ENTS, which is Xielei's method, which is in green. So you can see that ENTS and CMsearch are significantly better than HH-search and RaptorX. However, with respect to precision, when K is small, ENTS performs much worse than CMsearch, which suggests CMsearch is a better method. So the next question to answer is that, okay, although CMsearch can give you hopefully better homologs among the top K predictions, But does that necessarily lead to better protein structure models to be generated, which is our final goal? So in order to answer that question, we asked every single method to return their top K predictions. And we use modeler to generate one model for each of those top 10 predictions. And then we chose the best model among the top 10 for every single method. And here I'm drawing you the scatter plot where y-axis is always CMsearch and x-axis are the corresponding state-of-the-art methods. So I partitioned this scatter plot into 4 parts for you. So the gray part is the difficult cases, where none of the method can generate reliable structure model, which means TM score above 0.6. And the black parts are the simple cases, which means both methods can easily solve the problem. So the interesting part are the red and blue parts. So red means that the existing method failed to generate any good model in the top 10 model, whereas CMsearch sort of rescued this target by generating at least one good model among the top 10. And blue means that CMsearch failed, whereas the existing method succeeded. And if you take the average performance over all the 8,000 targets, then since this number is too big, any improvement will be averaged out. So, but still, no matter whether you use TM score or GDT score or root mean square distance to measure the top 1 model, CMsearch is always better than all the existing methods. If you look at these interesting red and blue regions, then you can see that CMsearch actually has dramatic improvement